It's my great pleasure to welcome David Krantz to UCSF. David is not a stranger. He did his postdoc with Robert Edwards in the 1990s, but prior to that he did an MD PhD at UCLA working with Larry Sapersky, where he was taught in the art and skill of Drosophila genetics and neurogenetics. And with Robert he learned about neurotransmitter transporters, and that combination of foundation has served him well, and ever since he's been using primarily Drosophila as a system to study how neurotransmission is regulated and how we can take advantage of that information to screen for drugs that may help with neuropsychiatric and neurodegenerative disorders. So with that very brief introduction, I welcome David.
Thank you. Thanks for coming. Let me put this on and drop some stuff. So the talk is in two parts, and half of you, I imagine, will think that the first part is kind of useful, but the second part is kind of obscure and ridiculous, and the other half will think that the first part is kind of stupid and cartoonish, but the other half is mildly interesting. But on the CME forms that you fill out, if you average all that together, you'll get a theoretical person who doesn't exist will think it's spot on. So here we go. We're going to talk about transporters and psychiatric disorders and then segue into flies. So, the educational goals. Do you guys fill out this little form? Yeah? We have to do it at UCLA, yeah. So, we're going to talk about psychiatric drugs. We're going to talk about the difference between ones that cause blockade versus efflux. And I want to tell you about these new drugs that target one of my favorite molecules. And finally, some work that we're doing on flies to understand something about how these drugs work and how we can get better drugs. So here's, by way of background, where most of the drugs that we use work. At the synapse, here's a vesicle with neurotransmitters and little blue dots. Calcium comes in through a channel. The vesicle fuses with the membrane. Transmitter releases, binds to receptors, and stuff happens. So how do you get the neurotransmitter in the vesicle, and how do you get it back in? That, of course, is by two different kinds of transporters, red ones and blue ones. The red ones are vesicular transporters, and they are responsible for putting, and you notice the neurotransmitter changed colors. So it's green now. When you go back and forth between old and new PowerPoint, it seems to do that. So the green neurotransmitters goes into the vesicle. It's released. Plasma membrane transporters, of course, terminate signaling and recycle them. Here's the libretto for the mammalian plasma membrane transporters. There's 3 GABA transporters, more if you want to be loose about it, 5 glutamate transporters, and I put the red and the blue separately. Neuromodulatory kind of stuff in blue, and fast neurotransmitters in red, and acetylcholine is somewhere in the middle. And then the vesicular transporters, they're different. And here we have the vesicular GABA or vesicular amino acid transporter, 3 for glutamate. A lot of these were cloned here by Rob Edwards. Is Rob here? Rob? No. And then 1 each for dopamine, norepinephrine, and serotonin. We're going to be talking a lot about VMAT, the vesicular monoamine transporter 1 and 2. And as a preview, remember that VMAT2 is in the CNS and VMAT1 is in the periphery. And that's important for a couple of the drugs we're going to be talking about that are starting to be used for treating tardive dyskinesia. Okay, so how do these things work? Here's the trick. It's a secondary transporter, so they don't do the transport themselves. They are parasites on the back of ion gradients. So here are ions. Here's blue dots are ions, and orange squares are neurotransmitters. And the blue really wants to go over here because there's not much of it probabilistically. And when it does that, it drags along the transmitter rather. So that's how vesicular transporters work. That's how plasma membrane transporters work. At the plasma membrane, you have a sodium gradient. Here comes some gratuitous animation. and the sodium gradient is driving the neurotransmitter into the vesicle. And you can see sort of alternating access where stuff binds here and then it opens. And that really is kind of how it works, an alternating access model where ions and substrate bind to the outside, a conformational shift occurs, and it opens so that the same molecules can come to the inside. And this is a cartoon showing the leucine transporter. That was one of the original ones for bacteria that was used to study this, but more recently there are crystal structures for the dopamine transporter and the serotonin transporter, and we've learned a lot about where the substrates bind and where the ions bind. Next question is, which cells are these things on? And as most of you know, for the monoamines, for dopamine and serotonin and norepinephrine, the transporter is right on the presynaptic neuron. Neurotransmitter comes out, it gets recycled and gets put back into the vesicle. On the other hand, other neurotransmitters, primarily glutamate and to some extent GABA, don't have that direct route. So when I was younger, this was pretty obscure. How many people knew this already? Can you raise your hand? How many people knew that glutamate does indirect? Okay, so this is not all in vain. So glutamate transporters, by and large, are not on the presynaptic neuron. Rather, you have to recycle them indirectly. The glutamate transporters are on glia, and who would design this? So glutamate gets turned into glutamine. Glutamine gets released through another kind of transporter. Glutamine comes back in to the presynaptic neuron and then gets changed back to glutamate. So it's inefficient, but I didn't design it. So, as most of you know, psychiatric drugs, a lot of psychiatric drugs, bind to the plasma membrane amine transporters, and in particular to DAT, NET, well, in particular for the SSRIs and for psychostimulants, SSRIs, SERD, and then for some of the psychostimulants, DAT and NET. Here's some of them listed here. And I want to highlight two critical differences, one in background and one is the educational goal 1A. The affinities are different, and I think everybody in this audience knows that the antidepressant affinities are different. Some bind NET, some bind SERT, but I think it's less well known that the psychostimulants actually have fairly different affinities too. So cocaine will bind all of them, but Ritalin or methylphenidate doesn't like to bind SERT. And the ones that we'll find out work as efflux agents also have this difference in affinity, where methamphetamine and amphetamines prefer the catecholamine transporters. And this can affect either both the efficacy of the transporter for particular symptoms, drug for particular symptoms, as well as the side effects. So, educational goal 1A. Any questions so far? It's a nice friendly group. People can ask questions. Yeah? What's blockade versus efflux? So who knows that? Who knows what the difference is? John, no? Okay, this is great. All right. So, mechanism 1, blockade. That's what we're all taught and what most of us are familiar with, where you bind the transporter with a drug, whether it's Prozac or duloxetine or dizipramine for those of us of a certain age, like myself, or cocaine and methylphenidate. So the binding site can differ, but the point is that it's blocking. So here's the same slide, and here is what happens when you have cocaine. You don't get the flip-flopping, and you just get the buildup of the orange triangles. Now they're triangles. The neurotransmitter that just stays on the outside, and if you have more dopamine or serotonin on the outside, there's more for signaling. Now, the binding site for cocaine is right at the substrate site. Not all drugs bind at the substrate site. For example, citalopram has 2 binding sites, and someday maybe we'll be able to exploit this to design better drugs. Meanwhile, let's learn about the second mechanism, which is efflux. And this is defined as reversal of the direction of transport. And the mechanism is really ridiculously complex and poorly understood, even though you think, hey, amphetamines, everybody knows how those work. They've been around for years. But we're still trying to figure out how they work. So the specific drugs that do efflux are amphetamine or methamphetamine, and therefore the catecholamines again, or MDMA or ecstasy. It's primarily for efflux of serotonin. So what am I talking about by efflux? Well, first, these things go in, okay? And it kind of makes sense because they kind of look like neurotransmitters. So here's amphetamine, here's methamphetamine, here's MDMA or ecstasy, and this kind of looks like dopamine. This just kind of looks like serotonin. So they go in the cell, and then they do something. So what do? They do something weird. They change the conformation of the transporter to something that's kind of like a neurotransmitter channel. So here you have the sodium neurotransmitter. Normally, sodium-coupled transport, it's going this way. What these efflux agents do is they change this thing to make essentially a hole in it, and the dopamine comes out in the other direction. Okay, how does that work? It's got to be something simple. It's not. So a lot of data, a lot of it from Aurelio Galli's lab at Vanderbilt, has shown that the mechanism of this is really complex. So the efflux agents indirectly activate a kinase cascade, CAMK2 for the aficionados in the audience. CAMK2 phosphorylates this, changes the conformation, and the neurotransmitter goes out, probably down its concentration gradient. Again, who would design this? Not me, but that's probably the way it works. So, amphetamines probably also act on vesicular stores. So they let the neurotransmitter out of the vesicle into the cytosol. Okay, so there's more out here, more can go out here. Well, how does that work? That is also complicated. And before we look at the potential mechanism, let's just look at some of the basic biological mechanisms of the vesicular transporters, which are different than the mechanisms of the plasma membrane transporters. So, they're on the inside. And because they're on the inside, they can't use the same mechanism to drive transport. So plasma membrane transporters use a sodium gradient. A lot of sodium out here, not much sodium here. There's not the same gradient across this membrane, so you can't use a sodium gradient. But, as luck would have it, there is a gradient available. There's lots and lots of protons inside the lumen of most intracellular organelles, and that goes for synaptic vesicles, it goes for the ER, it goes for Golgi, and there's different amounts of protons in each one of these organelles. There's a different gradient across each one. Gradient here is about 100-fold more protons here than out here. But you'll notice that they have to go in opposite directions. So before, the sodium's going in, and the neurotransmitter's going in, and that's called symport. Here, the protons are going out, down their concentration gradient, and they're shoving the neurotransmitters in against their concentration gradient, and that's called antiport. This is really good at cocktail parties, like you're talking to somebody, you go, hey, you want to hear about symport or antiport? It really works well. That's how I met my wife. So they move protons in the opposite direction as the neurotransmitter. So to make the gradient here, you need something else. You actually use an ATP-driven pump to shove the protons in here. So without a pump and without a gradient, things leak. So if you somehow disrupt this and you don't have any more protons in here, the neurotransmitter leaks out. And we're not really sure. Probably leaks out through here, but it's not clear whether it really leaks out through the transporter or through the membrane. So without a proton gradient, they leak. Amphetamines probably disrupt the proton gradient. How does that work? Again, this should be simple, but it's not. Here's what things look like under normal circumstances. You have a proton gradient going out. It's pushing the neurotransmitter in. There's two major hypotheses about how you break this. How do you disrupt the proton gradient so that the neurotransmitter will leak out? And the first hypothesis was from the 1990s by Dave Sulzer at Columbia, and he said these things work like chloroquine, which is a weak base, and they somehow get in to the vesicle, maybe through the membrane, and then they neutralize the protons, and so without the gradient, the neurotransmitter just leaks out. That's been around for a while. Still might be true, but Zach Freiberg, a couple of years ago, when he was with Jonathan Javich at Columbia, he's now at Pittsburgh, had another idea, and this is very attractive, and we collaborated with him, so it's particularly attractive, is that you don't use this mechanism. What you do is you use the transporter itself to break the gradient. So you take the drugs, you take the drugs, and you have a gradient of drugs down from the outside to the inside of the vesicle, and you make the transporter do antiport, but you're doing it with a different gradient. So usually the protons drive the neurotransmitter in. Here, the substrate, the neurotransmitter, is driving the protons out. And if anybody wants to talk about it later, we can talk about the data for that. It's very elegant. Either way, you disrupt the gradient, and the neurotransmitter leaks out. And if you have more neurotransmitter on the outside, maybe that potentiates efflux. Let's go to the second educational goal, which is talking about another drug, subclass of drugs, that binds VMAT1 and VMAT2, and which begs the question, who cares? Why would you care about VMAT blockade? Because people used to care. In the 60s, it was a really big deal because there was an antihypertensive drug called reserpine. Did anybody here ever prescribe reserpine? Okay. Has anybody here heard of reserpine? Yeah. Good. Yeah. Because when people started using reserpine, people got kind of logy, and maybe they looked depressed, and maybe these monoamines are really important for stuff. And then segue till today, and we know that monoamines really are important for a lot of stuff. In the 70s through 2017, there's another drug that binds to vesicular transporters called tetrabenzene, and people use that for Huntington's chorea. But actually, they used it for tardive dyskinesia too. Just there were no FDA trials, and it wasn't patentable, so it wasn't advertised a lot. However, in 2017, there was the first trial of a patentable pharma money-making drug called valbenazine or Ingreza, and on its tail came deuterabenazine or Austedo, to treat tardive dyskinesia. What is tardive dyskinesia? Some of you have seen a lot of tardive dyskinesia. Some have maybe not seen that much because the typical antipsychotics did it a lot, and the atypical ones didn't do it as much. But if you've ever seen it, it's very striking. These rhythmic chewing movements, clenching, lip smacking, protrusion of the tongue, and grimacing, and Nina Schooler and John Kane developed the Ames test to quantify this. And I want to highlight the idea, though, that it really isn't just the typical antipsychotics that do it. Unfortunately, there's a case every once in a while, probably some of your colleagues or you have given someone tardive dyskinesia, unfortunately, with an atypical antipsychotic. It does happen, less frequently, but it does happen. And Comprazine or metoclopramide, there's some reports of this actually giving tardive dyskinesia too. So what do we do about it? Now we can use an FDA-approved drug, one of two, to block the vesicular transporter, to deplete dopamine, and to decrease the symptoms by about 3 points on the Ames test if you use a higher dose of valbenazine. So I mentioned before that VMAT1 and VMAT2 are in different places. Tetrabenazine is more active on VMAT2, which is centrally, rather than VMAT1, and that's nice because you don't deplete the amines peripherally, so you don't get orthostatic hypotension. So the side effects are better for tetrabenazine derivatives than they would be for reserpine for treating this. Why do we have these two? Well, one is because it's patentable, and the other is there are a little bit better in terms of side effect profile and in terms of half-life. So this is clever. So if you put deuterium or a proton plus a neutron, rather than just a proton, at on some of the sites in tetrabenazine, cytochrome D6 doesn't metabolize it as well, and it actually changes the half-life by about half, and that's kind of convenient because this way you don't have to dose as frequently. And that's deuterabenazine, or Astato. Another trick you can use is to capture a subset of the isomers. So tetrabenazine has 4 different degradative products, and there's 1, 2 plus and a 2 minus. And what this company did is to take 2 of the isoforms, the 2 isoforms that are active, conjugate that to valine. And in that way, you decrease the number of isoforms that cause side effects, you maximize the number of isoforms that cause the positive effects, and you can sell it. Tetrabenazine itself is not FDA approved for this, but if you look back in the literature, you can see some case reports and think about it. Okay, we've done these 2 educational goals, going a lot faster than I thought I would. So I'll try to slow down, and if anybody has questions, please go ahead. We're going to now talk about, yeah.
Synthesis of molecule take place in the cytosol?
Yes.
Increase in that area?
Yes.
And do you know whether or not some of these drugs that are increasing efflux modify synthesis? That they might actually increase synthesis?
Yeah, the question is, do the drugs, whether tetrabenazine, amphetamines, any of these things act on the biosynthetic enzymes? In the case of dopamine, that would be tyrosine hydroxylase. In the case of serotonin, it would be tryptophan hydroxylase. I'm not aware of that happening, but amphetamines probably act on monoamine oxidase to some extent in blocking the degradation. So in the same direction as the direction that they're using to increase efflux, they also block degradation. And I omitted that for simplicity, but yeah, that's another way that they work. Any other questions before we launch into flies? Okay. So we're using flies, or we and others are using flies for a number of reasons, but today I want to talk about 3 reasons to look at how different types of release actually may have different effects on circuits, to do in vivo pharmacological screens, and you can decide for yourself whether that's a good idea or not, and as a cellular model for trying to understand how our current antidepressants work, which cartoons and Stephen Stahl aside, we still don't understand. So why use flies to do anything at all? One is scientific reasons. There's some great genetics. There's a lot of tools that really aren't quite in mouse yet, and definitely not in rats yet. And the other thing is, because they're small and because they don't live very long and because you can have thousands and thousands of in your lab, you can screen things. It's pretty hard to screen rodents. They're a little too expensive. Which brings us to the second reason why flies are nice. They're convenient, they're cheap, and most importantly, I don't write any IRB protocols. Is it reasonable to use flies? I think it is if you think about the right level at which you're using it. So there's very nice conservation at the level of molecules. The neurotransmitters, by and large, are very similar. The transporters and the receptors look very similar. The pharmacology is different, but the mechanisms are really conserved. And the cell biology is really similar too. There's differences in the shapes of the neurons where the dendrites come off. But if you look at synaptic transmission in flies versus mice or versus humans, it's pretty similar. So if you think about things at that level, I think we can learn a lot. The anatomy is totally different, and there's some genes that aren't there, like alpha-synuclein. But if you're trying to understand how do you change synaptic function and what does that do to circuit function, I think flies are a pretty good way to look at that. So the ways that we're doing that is, again, looking at how transporter localization affects circuits and behavior and how different vesicles might do stuff. Can we identify new drugs? And again, how do psychiatric drugs work? What happens when these extracellular ravines are increased? I'm going to focus on the monoamine neurotransmitters, dopamine, serotonin, noradrenaline, or in flies, something called octopamine. And I've drawn here what octopamine looks like, and here's a box. What's in that box? Nothing's in that box, but in mammals, there's an oxygen in that box. So noradrenaline is octopamine plus an oxygen here. I mean, what's one oxygen? You know, I mean, what's the big deal? So we think about octopamine as fly noradrenaline. It's for fertility, it's for locomotion. Dopamine in flies is used for reward and grooming. Serotonin for aggression, and what we're finding is it's probably used for vision as well, but that's a new thing. Drosophila have transporters, just like we do. They have fewer of each one of the types. So in mammals, there's GAT1 through 4. 4 if you count the betaine transporter. Flies have 1. Flies have 3 V-glutes. I mean, humans, mammals, sorry. Mammals have 3 V-glutes, flies have 1. And flies have flies have 1 kind of VMAT, we have 2. So it makes the genetics a little bit easier if you're trying to mutate things. You're not worried about, well, maybe there's going to be some compensatory something going on. What you'll notice here are these dots. Flies, as far as I know, and I tried, don't have an octopamine transporter, which is kind of a bummer, but we can use that later to do a drug screen where we want to eliminate that as a mechanism. So hold that thought for a couple minutes, and meanwhile, let's go back to talking about how we're going to use flies to study drugs and to study neurotransmission. Here we're back to our simple canonical model of neurotransmitter release. We have a vesicular transporter on here. Calcium comes in, fusion, neurotransmitter comes out, right? Well, the reality is really a lot more complicated, and this is the reality. So there aren't just synaptic vesicles. Dense core vesicles that most of you know from peptide storage also have amines in them. They have serotonin in them, and they have noradrenaline in them. What's up with that? Why do they do that? And not only that, but dense core vesicles that release serotonin, release noradrenaline, can be up here in the somatodendritic compartment. They can be down here in the nerve terminal. And so you have neurotransmitter release. You have serotonin release here. You have serotonin release here. You have serotonin release here. So somatodendritic versus active zone, synaptic versus extrasynaptic, synaptic vesicles versus dense core vesicles. And we really don't know what each one of these things do by and large. Maybe the one exception is psychostimulant sensitization. There's some papers from the 90s from Peter Kalivas that says that maybe cell body versus nerve terminal things are different for different aspects of sensitization. But other than that, it's like, well, stuff comes out, and we don't really know which stuff does what. So to answer that question, we're going to use flies, and we're going to focus on synaptic vesicles versus dense core vesicles to try to see, is there any difference between release from one type or the other type? And the way we're going to address that question is by moving the vesicular transporter around. So VMAT, in case of us, VMAT2 in flies, just Drosophila VMAT, is the same in each cell for each kind of vesicle. So in a serotonergic cell in your brain, VMAT2 is on the synaptic vesicles, and VMAT2 is on the dense core vesicles. In the noradrenergic cells, in your locus ceruleus, VMAT2 is on the dense core vesicles and the synaptic vesicles, and releasing in, we think, different ways. So the trick here is to disrupt the trafficking so we can change the mode of release. If we put more on here or more on here, we're going to release more from one of these and help address the question, does it matter or not? So, first thing is to see whether or not we can actually move the thing around. And so, ignoring a lot of in vitro work that took a number of years, we're just going to skip right to flies, and we're going to put a tag on the vesicular transporter, and then we're going to mutate this part here that changes its localization, and then we're going to ask, okay, if I mutate this part that from in vitro studies we think is going to change the localization, does it do anything? And it does. So we published this a few years ago. This is a complicated slide, but if I can walk you through it, look at black line here. Okay, this is the wild type transporter, and there's some on dense core vesicles, there's some on synaptic vesicles. This is a sucrose density fractionation experiment. We grind up fly heads in a tube, we put it in a centrifuge, and it goes to different densities. This density or that density for the different vesicles. And wild type, normal VMAT, goes to both places. If we start messing around with the trafficking domain, and the precise mutations aren't that important, but if we either chop off a chunk or we actually change one tyrosine to an alanine, we move it all over here, or a lot of it over here. So these two mutants are mostly in dense core vesicles. Yes?
I want to go back to the second your methodology.
Oh, yeah.
Take a whole lot of fruit fly heads where these vesicles are
Yes.
concentrated, and you mush them all up, and then you analyze the chemical substances that you find in the liquid phase.
The first part, yes. Sort of the second part. So we mush them up, Teflon on glass. We put them in a sucrose gradient on the top. They go down to their respective density. We fractionate the gradient. We take the fractions. So we could do what you said. We could look at the chemical, we could look at the dopamine and serotonin, but the concentrations are low. So what we actually do is Westerns. So we take a fraction from each fraction, and we run on a protein gel, we blot it, and then we use an antibody to that tag. I'm sorry I glossed over this. An antibody to that tag, which is the same tag in the wild type and the mutant, so we don't have to worry about differences in affinity to see where it goes. And so we have these blots where we say, okay, here's where the wild type is. It's in these fractions, and here's where the mutant is. It's in those fractions. Sorry I didn't explain that better. Any other questions? Okay. So it looks different. Does that mean anything? The way to ask that is to do something called genetic rescue, which means you get rid of all the VMAT, or any gene you want, and then you replace it with a transgene. So we have no more VMAT in the fly. Now we can put back normal VMAT. Or we can put back transgenes representing each one of these mutants and say, do they work or do they not work? Do they rescue whatever defects we see? Do they rescue storage of amines or not? Any guesses? The answer is yes and no. So the answer about rescue for some stuff is yeah, it does just fine. So if we look at storage here, we look at amine storage, and this is what you were asking about, but instead of on the fractions, we're just using the whole fly head, so we can get enough of in this case, dopamine to actually measure it by electrochemical detection on HPLC. Yeah, it looks okay. We rescue. We look at some behaviors that are defective when you knock out all exocytotic release of amines, and you look at male fertility, which doesn't seem so great when you can't release dopamine or serotonin or octopamine. But if you release it from vesicle type A, synaptic vesicles, or dense core vesicles, it doesn't really matter that much for that circuit, for the circuit that controls male fertility. On the other hand, for other circuits, I'm going to show you one here. For female fertility, it matters a lot. So here we have female fertility. Fertility, do they lay eggs or not? Fecundity is how many eggs they lay from a given animal. This is how many eggs a CS or Canton S, a normal wild-type fly lays. The null doesn't do very much at all. If we rescue with the wild-type form of the protein, okay, it's not perfect, but it's almost back to normal. But if we rescue with these mutants, they're almost infertile. And we can talk later about why we think this one is worse than this one, but it actually is, even though the molecular defect is a little bit less severe. So, very grossly, release from specific vesicles seems to be important for some circuits, maybe not others. This is a very simple idea, but to my knowledge, it's the first concrete demonstration of this very simple idea. And the reason it's relevant, I think, to psychiatry is because we don't really know where our drugs work. So there's transporters up here, and there's transporters down here. And if this thing is a drug, we're blocking transport here, and we're blocking transport here. Which one of them is really relevant for the relief of symptoms that we have? And people have made models that, well, this part is for the time course, and this part is for the actual efficacy. I don't know whether that's true or not. We don't know yet. But using these kinds of methods to move these things around, I think we're going to be able to get at that question. In other words, understanding the basic biological mechanisms of neurotransmitter release to understand how the drugs work eventually. We need new mutants to shift VMAT to synaptic vesicles. I don't have those yet. We have a nice circuit model that I'm not going to show you to study the mechanism. We're looking at the contractions of oviducts, contractions of the entire reproductive tract, and each one of the aminergic neurons that control that, but I'm not going to talk about that. And next, how do these drugs affect it? We can't do it on Amphetamine, but we can do it on other transmitter systems that are affected. Questions? Okay. Yeah, John? Yeah. So the question is, in flies or in mammals, does release from different kinds of vesicles have different effects on local inhibitory neurons? It's not clear yet. So the model that most people have looked at for antidepressants anyway, and thinking about where the somatodendritic release serotonin is acting, is based on local release and autoreceptors onto this. Is that me? Did I do that? I do have a phone.
Maybe,
You didn't confiscate it, so it's not like grade school. But you're right. All these local inhibitory neurons have receptors on them, and so, for example, pyramidal neurons in mammalian cortex that have serotonergic neurons project down into the Larvae onto local GABAergic neurons that have serotonin receptors to form a local feedback loop. So, and we don't know which kind of vesicles are actually maybe acting on those local GABA inhibitory neurons. Similarly, we don't know which type of release is important for the inhibition or the excitation of serotonergic receptors on these pyramidal neurons. And what's kind of interesting is that some of them have receptors with opposite effects. or at least on paper, opposite effects that do one thing in one part of the cell, another thing in another part of the cell. So it's a great question, and I hope we can answer the question someday. Yeah. Right. We haven't tried that. It's an interesting idea. So you're saying increase synthesis to see whether or not you can even the playing field by just making everybody fill up to the max? I don't know. So there's one group, Gonzalo Torres, who says that tyrosine hydroxylase may actually physically interact with the vesicles and dopamine transporter. I think the data is okay. I think the data is better for glutamate, where there's actually a local synthesis through a different pathway, through alpha-ketoglutarate, and there probably really is local synthesis specific for the vesicles. And there's some even recent data saying that you may get a little oomph, you may get a little boost just before these things are released for them to get filled. Acetylcholine, there's another phenomenon like that called second stage loading, where just before the released stuff comes out. So it's an interesting question. I don't have the data for amines to answer it, but it seems you're right that you should at least be able to overcompensate at some part in the pathway to overcome these effects. Any other questions? Is there fertility and these things? There is noradrenaline, and there's amine regulation in our reproductive, well, not mine, mine too, my reproductive, male and female reproductive tracts. We haven't looked at, I don't think anybody's really looked at fertility in a VMAT loss of function mutant in the mouse. It's an interesting idea. They're not very healthy, and for the most part, they die very early, unless you actually feed them amphetamines. That was an experiment done by Rob Edwards in the 90s. So we wouldn't be able to tell whether or not it worked or not. I don't know. And for these guys, you mean for the trafficking mutants? Between fertility and schizophrenia, there's differences in fecundity. It's so complicated that I'm not sure you could actually parse out what was because of a local physiological effect versus a more global effect on the behavior of the people. Interesting question, though. Okay, 2B. We're going to try to use flies to try to find new drugs. So backing up, how are our current drugs found? By accident, by and large. And once they're found by accident, by saying, let's make another one that does the exact same thing but differs by one carbon, and we'll call it something else. So wouldn't it be nice to use a more rational method to design drugs or to discover drugs? And wouldn't it be nice to use unbiased screens to do that? And I want to highlight where those unbiased screens should be looking. So lots of antidepressants block reuptake. The older ones block degradation. There's receptor agonists, mirtazapine. And amphetamines cause efflux. But to my knowledge, there are no antidepressants or anti-anxiety agents that will increase storage and decrease storage with reserpine and tetrabenazine, but you can't increase storage. And there's nothing that increases exocytotic release. So if reserpine causes people to be logy and maybe depressed, and amphetamines help with ADD. Could we make a drug that somehow makes this work better or makes this work better? But if to do that, you really need all the machinery here, because it's really hard to make a drug make something work better. So if we make things work better, it's probably going to be down here, making a regulatory thing work worse. So if we inhibit an inhibitor, then maybe this will work better. But it's probably going to be indirect. But if it is indirect, you really need the whole synapse. You need all the parts there. And I don't think it's going to be as easy to do in a reduced system. And that's my justification for using a whole animal, for using modifier screens in a whole fly. And the trick here is to use something called a sensitized background. We mess up the fly a little bit to make them sensitive to drug effects, and then we screen for modifiers of that effect. And the modifiers can be drugs or they can be genes. Very briefly, here's a proof of principle. So fly larvae depend on octopamine to move around. If you give octopamine, somehow they eat it and it goes to their central nervous system. I don't know how. Sometimes you give the wild-type larvae, and you really can't tell if you give them octopamine, if anything's happening at all. Notice the y-axis here, where the number of lines crossed per minute is really high. Now look over here. We have a mutant, happens to be VMAT, where they can't crawl very well. Notice the y-axis here is really, really low. And even if you rescue it, it's still really, really low. But the difference is really easy to detect, okay? So the difference between this and this, you can see the difference, okay? So the sensitized background gives you a way to detect stuff that you wouldn't ordinarily be able to detect. And here we're using it with a neurotransmitter, but we applied that to drugs. So we gave 1,000 drugs to larvae, and then we looked at whether or not they would move or not move, and Hakim and a small army of undergraduates did that. And I'm not going to show the movie, but basically they start here, and if they move, they go over here. We found some, and some other genetic tests showed that they act presynaptically, so we can knock out the, I'll skip over the details. And the trick is that because we used a circuit where there is no transporter, there is no octopamine transporter, we were pretty sure that they weren't acting at the level of a transporter. So we think this is acting somewhere around here, and that's nice because there aren't any drugs that act somewhere around there. And in fact, one of the ones we found was very surprising. It's called dicarbazine. It's an alkylating agent. It's used as an anti-cancer drug. And it's like, oh, that's so, oh, you failed. It's wrong. But it turns out that if you give dicarbazine, people start throwing up because their serotonin blood levels go up. So I think this is a loose proof of principle. So the next step will be to try to use additional tests, biochemical tests, to understand what the drugs actually do. It's been really hard. So instead, what we're going to do is go back and redo these things using a genetic screen to identify concrete targets so that we don't have to guess on what the target is, and that's ongoing. So in the final few minutes, I'm going to figure out how SSRIs work. And this is a collaboration with Mark Fry's lab at UCLA. Okay, full disclosure, I haven't figured it out yet. But I think we have a system that's going to give us a first step. As you know, lots of antidepressants block transporters, including the noradrenaline transporter, serotonin transporter, and then you get this stuff out here. You get more neurotransmitter extracellularly. We don't know what happens after that. So you have lots of neurotransmitter out here. We don't know what happens acutely. We don't know what happens chronically. So there's going to be a circuit effect. There's going to be some systemic effect. That's not just one cell. But as a first step, it would be really nice, or I would really like to know, what happens in the first cell that sees this signal that's different? And we need a model. So to explain a little bit more clearly, I want to know what the transcriptional changes are in the first cells that see a difference in serotonin extracellularly. And the problem is that the way most people currently look at this is you take a hunk of brain and you grind it up and you look at what happens with the RNA species in like 100 or 1,000 different kinds of cells. And I understand bioinformatics is really, really powerful. I don't think it's powerful enough. So in my opinion, I think you really need to be more fine, do a more fine analysis, and to look at individual cell types where you can actually detect what's happening in one kind of cell at a time with one kind of receptor. So you can do that maybe if you can isolate single cells. But the other way to do it, and this is a trick we're using, is find a system where there's a lot of cells that are exactly the same. And it just so happens that the fly visual system is like that. So fly eyes have 750 light apparatuses on each side called an ommatidium. And each one of these clusters of photoreceptor cells projects down to a first ganglia called the lamina. This projects down to the next ganglia called the medulla. These are really important for movement detection. But the key point here is that each one of these things has to work the same, otherwise the fly is not going to be able to tell that this part of the visual field is the same as that part of the visual field. And each one of these things projects down to the exact same type of cells. So in each fly optic lobe, you have 750 copies on each side of about 25, 30 different kinds of cells. So what we didn't know is whether any of them have serotonin receptors or not. So we've spent the past couple of years building a system to ask this question. We've mapped the localization of serotonin receptors, there's 5 of them in the fly, to specific cell types. And all I'm going to show you here is the bottom line that, oh look, here's the cell. We know that this is the L2 type cell. We know this is a T1 cell. And we can see through genetic tricks that 5-HT2B is in this one, 1A and 1B are in that one. But we really need to know whether they do anything. So the next thing to do was to see whether or not these cells respond to serotonin. So what you can do, and this is in collaboration with Mark Fry, not Fly, you can mount the fly like this, and if you're very good, you can tear off its cuticle just above one of the eyes. It's very small. I haven't tried to do this, but there are people in the lab who can do it. And if you do that, and then if you use a fancy microscope with 2 photons rather than 1 photon, you can see images like this if you look at calcium probes. And if you look at calcium probes, that'll tell you, is the cell working? Is the cell not working? And then what you can do is give it serotonin and say, does anything happen? And what you predict, this is the part where it's too complicated, where you predict that serotonin 2B is coupled to GQD11. You predict calcium increases when you give serotonin, and you do see a calcium increase when you see serotonin. So now we have a system where we've mapped these serotonin receptors onto specific cells. We know they respond to serotonin, and now we want to build a circuit diagram. We've partially filled it in with how these things are connected to each other. But most importantly, we've developed a method to isolate them and interrogate their transcriptional profile. And then that gives us the ability to see what happens to their transcriptional profile when you start changing extracellular serotonin. So in brief, you have a so-called driver. You express glowy GFP in one cell type. You dissect the fly brain. That's easier than taking off a piece of skin. You dissociate the cells. You use fluorescence-activated cell sorting, and you get green cells that you sequence. And we've done that. Works great. It's reproducible. We get some genes that we didn't really expect to be there. And now the next step is after the baseline that we have to inhibit SERT. And those experiments are ongoing using pharmacological probes as well as genetic probes to inhibit the transporter. The different people that did this work, the trafficking was done by Anna Gregoric, drug screen by Hakim Lawal, Kathy Meyer, Geschwing, and Maureen in my lab, and Ben Hardcastle and Mark's lab did the serotonin stuff, and some people gave us money. Thank you very much. Questions?
Now a lot of people actually see that you can deplete other neurotransmitters other than mono gives you unless you're pure animals. So I was wondering if you had a sense of like physiologically these type of matters and how specificity and whether or not drugs like Reserve actually what they're really doing is like blocking or decreasing GABAergic resistance.
Really interesting question. So a very prominent paper from Bernardo Sabatini that said that VMAT can store and release GABA. Might be true, but what's definitely true is that there's lots of cells in the nervous system that will express both VMAT and a vesicular glutamate transporter, or both VMAT and a GABA transporter, and similarly acetylcholine transporter and something else. And it's possible that there's co-release. It's also possible that one regulates the other one. So in addition to functioning as transporters, these things are also ion channels. So there's a story from Rob and other people that says that glutamate actually, the glutamate transporter actually regulates how much dopamine or how much serotonin is in these cells. So you're absolutely right. There's not one-to-one specificity for what each one of these drugs is doing. And I think it's going to be important to go back and to see whether or not they do change anything in places that we previously didn't expect them to change.
I want to thank you for saying periodically it's really much more complex than this. And just for our trainees, you had lists of neurotransmitters that were like less than 10 in the columns. How many neurotransmitters do you think there might be?
There aren't that many classical neurotransmitters, little ones that are the size of amino acids, like glutamate, GABA, dopamine, serotonin. There are lots and lots of peptide neurotransmitters. And those are longer than one amino acid. Those are more like 5 to 30. So enkephalins are like 5, endorphin is 30. Those actually don't use transporters to get into the vesicles. They go in through a different pathway. So the actual small ones, there aren't that many. I left out adenosine, ATP, and histamine, but there's not like dozens and dozens more of the little ones. And does that answer your question, Anna? We cut the fly in half, literally. So for the circuit that we're using now, we're looking at a cluster of 9 octopaminergic neurons that are in the ventral nerve cord. They project down to the reproductive system. Central nervous system, that would be a really hard thing to look at, but we can cut the ventral nerve cord away from the peripheral organs, and that's, at least in that circuit, the way we're doing it. Does that answer your question? No, we don't cut the fly in half when we're doing the behaviors. So you're right. We can't tell central versus peripheral effects when we don't cut the fly in half when they're actually behaving. But that's actually why I'm focusing on this really simple circuit, because in the brain you have all these inputs and you don't really know what's going on, and you don't really know what's going on peripherally versus centrally. So yeah, I think for behavior, it's going to be really hard to tell. And to find circuits where you actually can parse out those things, at least to model the very simple effects, is going to be, I think, important. Thank you very much.